All right, let's talk about a challenge that is just mind-boggling. Imagine trying to find one specific person in a city of trillions. That's kind of what scientists are up against every single day when they try to find one specific protein. So, how on earth do they do it? I mean, just think about it for a second. Right now, inside your body, there are countless tiny protein machines. They're carrying oxygen, they're digesting your lunch, they're fighting off viruses. They're literally the microscopic engines that make life happen. So, if you want to understand a disease or figure out how to design a drug to fix something, you need to isolate and study just one of those protein types. But how do you possibly pull that one single needle out of that gigantic biological haystack? Well, let's dive in. Our quest starts inside a single cell, which, let me tell you, is an incredibly crowded and chaotic place. You see, when scientists crack open cells to look inside, they don't get some neat organized library. Oh no, they get what's called a crude extract. It's basically a messy, chaotic soup with thousands of different kinds of proteins all bumping into each other. And our target protein, it's in there somewhere. Okay, so how do you even begin to sort through that mess? Well, we use this brilliant set of techniques that act like incredibly specific smart filters. Welcome to the world of chromatography. You know, the core idea here is actually pretty simple, but it's super powerful. We take our protein soup, that's the mobile phase, and we pour it through a column that's packed with a special material, the stationary phase. As all the proteins travel down, some of them stick to that filter material more than others and get slowed down. Others just zip right past. And that difference in speed is what starts to separate them. And this is where it gets really clever. We can choose our filter based on a protein's specific properties. We can sort them by their electrical charge, or by their size, or even by a really unique shape that only they can bind to, kind of like a lock and key. Each one of these methods is another step that gets our sample cleaner and cleaner. So let's look at one of these methods, because it's a little bit counterintuitive. You'd think in a race, the small, nimble things would be faster, right? Well, not here. In size-exclusion chromatography, the column is filled with these little porous beads. The tiny proteins get stuck exploring all the little nooks and crannies inside the beads, so they take this long, winding path. But the big proteins, they're too big to fit inside, so they just flow right around the beads and shoot out the other end first. The big guys actually win the race.
So, we've filtered our sample a few times. We're pretty sure we're getting close to isolating our target protein. But now comes the moment of truth. How do we actually see if all our hard work has paid off?
I mean, we can't just look into the test tube. We need a way to visualize the proteins at each step of the process to see if we're really getting rid of the junk and concentrating our target. And this is how we do it. It's a technique called SDS page. Basically, we load our samples into a gel, think of it like a slab of Jell-O, and we apply an electric field. Now, a special detergent called SDS coats all the proteins and makes them negatively charged. That means they all want to race toward the positive end. The only thing separating them now is size. The gel is like a dense forest, and the smaller proteins can wiggle through it way faster than the big, bulky ones. And here it is, the results. The lane on the far left, that's our starting chaotic soup. Just a big ol' smear of hundreds of proteins. Now, each lane to the right shows our sample after another round of filtering. Just watch. See how the number of bands gets smaller and smaller until we reach that final lane on the right? We've gone from a total mess to a single, beautiful, clean band. That's our protein. So the takeaway here is really simple. On one of these gels, the goal is to get rid of all that noise. You want to go from a messy smear to as few bands as possible. Fewer bands means higher purity, plain and simple. The visual evidence is awesome, but you know how it is in science. We need numbers. We need a way to actually calculate a purity score to prove we've really done it. This is where a cool concept called specific activity comes in. If our protein is an enzyme, meaning it has a specific job to do, we can measure how well it does that job. Specific activity is just the total amount of work being done, divided by the total amount of protein in the sample. So, as we get rid of all those other useless proteins, that number should go way, way up. And this table shows the whole journey in numbers. Just watch the total protein column. We start with 10,000 milligrams, and we methodically throw out the junk protein until we're left with just 3 milligrams. But now, look at the specific activity column. It starts at just 10, and with each purification step, it absolutely skyrockets. And that final number is just staggering, 15,000. We have massively increased the concentration of our target. We're talking about a 1,500-fold increase in purity. The numbers confirm exactly what the gel showed us. We found our needle in the haystack. So we've gone on this epic scientific treasure hunt. We've filtered and sorted and raced these proteins. But what's the payoff? Why go to all this trouble in the first place? Well, this is why it matters so much. Once a protein is pure, we can study it all by itself. We can figure out its exact 3D shape, understand its role in diseases like Alzheimer's or cancer, and even design drugs that specifically target it. Seriously, isolating a protein is often the very first step to unlocking the most fundamental secrets of biology. Every single time a protein gets purified, it's like finding another piece at the puzzle, another clue to this incredibly complex machine we call life. It really makes you wonder, doesn't it? What amazing discovery will the next one unlock?